and questions of personhood arise. So that's maybe something to discuss later. But I want to just now stress to you that the reason people made these stem cell lines is because they were interested in understanding what is it about a stem cell. So what's happening in the nucleus? What's happening with the genes in these stem cells that they can maintain their stem cell fate indefinitely? And what happens when those cells do get triggered to differentiate, to form other tissues? What's happening in those cells to those genes? And so now I want to switch to nuclear reprogramming because that's going to, these are all going to come back together again. But this is the second aspect of what I want to tell you. So I just emphasized that we know that stem cells have this pluripotency, and we know that as cells differentiate, they lose that pluripotency. And so the question that scientists were really intrigued by was what is happening in those stem cells as they do become differentiated. And so one obligate thing that happens is that they lose their totipotency. Now, scientists now, developmental biologists, we now really do understand the basic idea of what happens to cells, to the genes in embryogenesis as cells are differentiating. But at the time when these studies were first being done, there were various hypotheses out there. So one hypothesis is that as a cell starts undergoing its differentiation and commits to a specific fate, whether it's a skin cell or a bone or a muscle, then perhaps all of the genes that are not required for that fate are lost somehow. And that would then cause that cell and that DNA in that cell to be committed and irreversibly committed. The other idea was that maybe all the genes are still there.